Hello and welcome back to IB Bio Made Simple. Today we're going to talk about topic 11.3, which is the kidney and osmoregulation. As always, we start with our essential ideas. You can see here, all animals excrete nitrogenous waste products, and some animals also balance water and solute concentrations. So as we know from our 7 major functions of life that we talked about way back in 1.1, maintaining a constant internal balance, homeostasis, is one of the functions of life. All organisms need to maintain a constant internal balance so that their cells can be healthy. And also, all organisms produce excess waste products by the processes of metabolism, and so there needs to be a way to get rid of those. So animals produce nitrogenous waste, that's waste that's composed of nitrogen in their metabolic processes, and they need to get rid of those, otherwise they can act as a poison to them, and they can build up and they can cause some problems. So all animals have to get rid of their nitrogenous waste, and they have to maintain that constant internal balance. And also, the form in which nitrogenous waste is excreted reflects evolution and ecological niche occupied by the animal. So not all animals excrete their waste the same. A fish excretes its waste differently than a bird, which excretes its waste differently than a human. And we'll talk a little bit about that. So here are our understandings, and we are only talking about part of this topic today. So we're not talking about all of the understandings, but we're talking about these first 4. So animals are either osmoregulators or osmoconformers. The Malpighian tubule system in insects and the kidney both carry out osmoregulation and the removal of nitrogenous waste. The composition of blood in the renal artery is different from that in the renal vein, and then how the ultrastructure of the glomerulus and Bowman's capsule facilitate ultrafiltration. And then we're also going to talk about this one down here. We're going to talk about understanding number 9, which is that the type of nitrogenous waste in animals is correlated with evolutionary history and habitat, and that's what I just referenced. And then we have our applications and skills. We're not going to talk about the applications today, but we will be addressing both of these skills. So drawing and labeling a diagram of the human kidney and annotation of diagrams of the nephron. We're not going to complete these because we're not going to make it all the way through all the structures of the kidney today, but we'll touch on them. So to start, we have excretion definition. So excretion is the removal from the body of the waste products of metabolism. So it's specifically the waste products of metabolism. So we don't consider defecation as excretion because feces is not the waste product of metabolism. Feces is undigested food. Okay, well, excretion by definition is waste products of metabolism. So in humans, that would be urine, which is so eloquently illustrated by Calvin here. So much of metabolic waste that's produced is in the form of nitrogen. We need nitrogen. It's part of some really important components for animals, specifically proteins and nucleic acids, both of which we cannot live without. But we don't necessarily need the nitrogen in those. So we have to find a way to convert that nitrogen into a form that we can then get rid of. So there's a couple of different ways to do this, and they are the 3 main categories we talk about, as I kind of indicated earlier, are going to be fish and amphibians, birds and reptiles, which are much more closely related than we would commonly think, as we know from back in topic 5.3 when we talked about different classifications, and then terrestrial animals. And terrestrial animals and mammals, more specifically, is where humans fall in. So if we start with fish, fish obviously have constant access to water. They live in water, so there's no need to make their excretion less toxic because it goes directly into the water and is watered down by it, and it's made much less concentrated. So they can excrete their waste as just straight up ammonia. And ammonia, any of you who have ever cleaned a bathroom will know that ammonia is a very strong chemical. And it's the simplest form. So down here we have the formula for ammonia. It's just NH3. So it's a really simple format. It's really easy. Doesn't take a lot of energy to make, and they can put it right in the water. Mammals, so that'd be us, including some other amphibians, some sharks, and some fish actually as well. We excrete our waste as urea. So we create ammonia, but we can't just excrete pure ammonia. Instead, we excrete urea. So it's a little bit of repackaging as less toxic molecules. Another reason for this is that to excrete ammonia, it requires a lot of water use by the organism. So while that's not really an issue for aquatic animals, mammals, for the most part, we want to retain our water. We don't just want to be getting rid of our water. So by converting that ammonia to urea before excretion, we can lose a little bit less water when we need to do our excretion. And then reptiles and birds go one step further, and they turn their their ammonia into this compound called uric acid. And uric acid requires more metabolic energy to make. You can see that it's a much more complicated chemical, but it is less toxic and it requires very little water to flush from the body. So when they excrete their metabolic waste, they are maintaining as much of their water as possible. So those are kind of the three main ways that we can balance how we get rid of nitrogenous waste. So ammonia requires the least energy but loses the most water. Urea, a middle level of energy and a middle level of water. And uric acid requires the most energy but the least water. So it's kind of a balancing act, and it depends greatly on the needs of that organism in its environmental niche. So if we look back at understanding 1 here, we have 2 different categories that animals can fall into. They can either be osmoconformers or osmoregulators. And hopefully you'll recognize the prefix osmo. Back in topic 1.4, we talked a lot about osmoregularity and osmoregulation. We talked about isotonic solutions. We talked about hypotonic solutions. We talked about hypertonic solutions. So osmo refers to the essentially the concentration of solutes inside the body. And then we have conformers and we have regulators. So osmoconformers kind of tell you what they do in their name. They conform to their environment. So they maintain internal conditions that are equal to that of their environment. So the inside of their body is just going to have the same osmolarity as the outside of their body. This is helpful to them in that it minimizes the water movement that actually goes out of their cells. However, a disadvantage is that they cannot optimize their internal conditions. So if they are in an environment that does not have optimal conditions, there's not really anything they can do about it. Most of these osmoconformers are marine invertebrates like starfish. So they will take in water, there will be water loss through skin, and essentially they have no direct control over the internal regulation of some of these ions. So we have sodium, we have potassium, we have chlorine down there. They just kind of go with the flow, and whatever is in the water, that's what turns out to be in their body. And then we have osmoregulators, which tightly regulate their body's osmolarity. So this is kind of more of what we would expect to see with homeostasis. So their body's osmolarity stays constant, irrespective of the environment. So it doesn't matter what the environment they're in, they're going to keep a constant internal environment. And so this is where the kidneys come in handy. They play a large role in osmoregulation because they regulate the amount of water that is lost from the body. So they regulate the amount that's reabsorbed, and we'll talk about that later. Actually, probably next time. But they regulate the amount of water that's in the body. Disadvantage of this is that active regulation, anytime anything active, that's going to cost ATP. So these organisms, these osmoregulators, have the expense of ATP, but the benefit of having a constant internal environment. So again, it's a trade-off. Osmoregulators in the animal kingdom are much more common, and humans would fall under that osmoregulator category. So, understanding 2 is the Malpighian tubule system. So essentially what this is it's a simplified version of a kidney that can be found in insects, not in plants. That would be very odd. That can be found in insects. So what we have here is, instead of having kind of the complex internal digestive system that you'd expect to see in most animals, insects have kind of this simplified continuous gut. So there's not really, you can't really see any distinct organs in there. Kind of have a continuous tract. And basically, in the stomach, there are these structures. So these greenish looking structures are the Malpighian tubules. So I have epithelial cells that are actively transporting salts and uric acid into the tubule lumen from the hemolymph. And then water enters that by osmosis. So hemolymph is the fluid in insects that is sort of like blood. They don't have a circulatory system. Their hemolymph is the fluid that functions as blood, but it does not stay within tubes. So our blood, as we know, is always in vessels. In an insect, they have what's called an open circulatory system, and so it's all just kind of circulating in the interior of the insect's body. Basically, the interior of the body is just bathed in this fluid, and that fluid is called hemolymph. And so the tubules will absorb, they'll actively transport salts and uric acid into the tubule lumen from that hemolymph. So they will take salts and uric acid and some water out of, they'll take salts and uric acid out of the hemolymph, and then water follows by osmosis, because if I take salts and I put them into the tubules, then that increases the concentration of the tubules, and water, as we know, likes to flow towards high concentration. So it will go into those tubules. From the tubules, the fluid will move into the hindgut, which is sort of like the intestine in us, where salts and water are then going to be reabsorbed. So salts and uric acid come in here, water follows, they move down, they come into the intestine area, and then the intestine will reabsorb some of those salts and water. So I'm left with mostly that uric acid is going to be left in here. It's going to mix with the feces, and it's all going to be kind of excreted together. So it's a pretty simplified way. Their uric acid here is their nitrogenous waste. They actively transport it into the tubule. It moves down with the help of some water, and then when it gets to the lower intestine and the hind gut, then the water is removed, and the uric acid, that nitrogenous waste, can be excreted along with the feces. So in this case, we have an example where they're combining their excretion and their defecation. So insects, pretty simplified strategy, but effective nonetheless. In humans, we have a bit more of a complex system. So first, major blood vessels that we have here. We have the vena cava here moving up. We have the renal artery coming from the aorta moving down. And so in this case, we know that the vena cava, from our study back in topic 6, we know the vena cava is bringing blood back to the heart, and the aorta is bringing blood away from the heart back to the body. So this works kind of opposite in the kidney. So in this case, the aorta comes down in blood vessels, and it turns into the renal artery here. And in this case, the artery, although we know that arteries go away from the heart, in this case the artery is going toward the kidney. So the artery brings blood into the kidney, and that is so-called dirty blood. It is uncleansed blood. It is blood that has those nitrogenous wastes in it, so it needs to be filtered. And the renal vein is taking clean blood out, so that is filtered blood where the nitrogenous wastes have been removed. And we can see down here as they come in, so the renal artery comes in, it kind of splits. We have these little lobes. The little lobes come back together into the renal vein, and it will take the clean blood out. We also have the ureter here, which is what's going to take the urine away from the kidney. Everything that was filtered out of the blood gets taken through the ureter and gets stored in the bladder until it's ready to be excreted. And then we have a couple of components of our kidney. So we have the outer area, which is called the cortex, the medulla, which makes up these lobes you can see, and then the central region is the renal pelvis. And all three of those are going to become important as we move on. So that is a general structure of the excretory system and the kidney. So from the kidney, the waste products that are filtered out will go down the ureter. They will come down to the urinary bladder, and then when they're ready for excretion, they'll go through the urethra and out of the body. So the kidney is the way that the body is able to filter blood and balance osmolarity. So the kidney is a extremely important organ in humans. So again, we have these three areas. We have the cortex, which is the outer area. We have the medulla, which is the middle area. And then we have the pelvis, which is the most central area. So they all have different roles. The cortex is for ultrafiltration and selective reabsorption. The medulla is where we reabsorb water so that we don't lose too much water. And the pelvis of the kidney contains the collecting ducts that are going to collect the urine that has been created and pass it on to the ureter. The ureter, as we mentioned, is going to carry that urine down to the bladder. And then we have the two major blood vessels. We have the renal artery that's coming into the kidney, carrying unbalanced blood, and we have the renal vein that's leaving the kidney, carrying balanced blood. So we talked about earlier, one of the understandings we talk about is that the composition of blood in the renal artery is different from that in the renal vein. And that makes sense because the goal here is to filter the blood. So you're going to go from unfiltered blood in the renal artery to filtered blood in the renal vein. So blood in the renal vein, when compared and contrasted with the renal artery, blood in the renal vein is going to have no change in proteins. Proteins are not filtered out in a healthy kidney. Blood in the renal vein is going to have less urea and toxins. It is going to have less oxygen. It is going to have more carbon dioxide. It is going to have fewer salts and ions. It's going to have less water, and it's going to have less glucose. So a couple of those may jump out at you, specifically oxygen, carbon dioxide, and glucose. And as we can see down here from our starred area, the reason for those changes is just cell respiration. So the kidney is made of cells and needs to do cell respiration, and cell respiration uses oxygen, so I'm going to have less of it on the way out. It uses glucose, so I'm going to have less of it on the way out, and it produces carbon dioxide, so I'm going to have more of it on the way out. So those aren't really due to the filtration process, they're just due to the fact that the kidney is made of cells, and cells have to do cell respiration. And then I'm going to have my toxins, which are my undesired wastes that are removed from the blood. And I'm going to have a change in salt and water concentrations because we are osmoregulators. So the kidney is going to balance these. So the degree to which the difference in salt and water between renal vein and artery is going to depend on how balanced the blood going in is. The more balanced it is going in, the more similar it'll be going out. And if it's really unbalanced going in, then you're going to need a lot of changes to take place. So those are the changes that you can expect to see between the renal vein and the renal artery. So balancing blood, the overview here is that we have ultrafiltration occurring. Then we reabsorb glucose, salt, and water. In ultrafiltration, basically everything is taken out of the blood, but then we want to reabsorb some of that, so we want to reabsorb glucose, salt, and water. We then have osmoregulation take place in the medulla and then excretion in the pelvis. And so this is a diagram of the nephron. This is the functional unit of the kidney. So this is the actual tubules that you can see over here span from the cortex to the medulla. This dotted line kind of indicates everything above that dotted line is cortex, everything below that dotted line is medulla. And so the blood comes in here, it gets filtered out, and then that filtrate passes through this long series of tubules and undergoes many changes and then finally becomes urine. So a couple of structures that we need to know about with the kidney. So this is where your annotations of diagrams of the nephron. So a couple of things that you need to know how to label in the nephron. So first we have the renal capsule. That's where ultrafiltration occurs. The glomerulus within that is this kind of capillary-like network that delivers blood to the renal capsule. So the glomerulus is delivering blood, the renal capsule is doing ultrafiltration. From there, we have the proximal tubule, which is where selective reabsorption occurs. From there, we go to the loop of Henle, which is where we have osmoregulation taking place. From the loop of Henle, we come back up and we go to the distal convoluted tubule, which is where toxins are secreted into the urine. So they are removed from the blood and put into the urine. And then we have the collecting duct, which collects the urine, multiple nephrons. So you can see these branches are multiple nephrons all come together into the collecting duct. And that is what will turn into the ureter and deliver, well, it'll deliver urine to the pelvis and then become the ureter. So here we have our final portion of the notes that we're going to talk about today, which is ultrafiltration in Bowman's capsule. So we have Bowman's capsule here with the glomerulus on the inside of it. So Bowman's capsule is kind of this paler orangish shade, and then the glomerulus is kind of the pinker network of vessels you can see in there. And so we have the afferent vessel, the afferent arteriole, which is coming into Bowman's capsule, and then we have the efferent arteriole, which comes out. So afferent approaches, efferent exits. And you can notice there's a sizable change in the size of the arterioles. There is much larger blood vessel in the afferent arteriole than the efferent arteriole. And because there is a larger blood vessel in the afferent arteriole, that means the pressure is smaller. So I increase pressure of the blood when I'm coming out. And so there's really high pressure inside of Bowman's capsule. And basically what that does, the reason for that, is that high pressure forces water and the blood contents to leave the capillary walls and go into Bowman's capsule. And that can then be captured by Bowman's capsule and continue into the nephron. So essentially the capillary wall here has pores in it that allows fluids to move through it. But the basement membrane allows only smaller molecules to be filtered. And so I have the interior of the blood here, I have Bowman's capsule over here, and in ultrafiltration all of these things are moving from the interior of the blood vessel into Bowman's capsule. So I have glucose, I have amino acids, I have urea, I have water, plasma proteins, red blood cells, and blood platelets, all of those in my blood vessels, but obviously I don't want the red blood cells and the blood platelets to leave. So I, between the basement membrane and the foot projections on the podocyte cells, they kind of act like filters, so only this smaller stuff can make it out. But plasma proteins do not get filtered out, and blood cells do not get filtered out. So an IB question could ask you to explain the process of filtration. I would pause here, try to jot down as much as you can remember from that last slide. And here is the mark scheme for that. So it occurs in Bowman's capsule, it occurs in the kidney cortex. The afferent arteriole has a larger diameter, which creates a higher pressure inside the capsule. Some smaller molecules are forced out through basement membranes. The molecules in the filtrate include these. Plasma proteins, cells, platelets are too large, so they remain in the blood, and the glomerular filtrate is then transferred to the nephron. So that is the process of ultrafiltration. That's the first step in the kidney's filtering of blood, is to take basically everything out of the blood so it can sort the good stuff from the bad stuff. So that's where we're going to stop for today. I hope that you learned a lot, and we are going to continue our examination of 11.3 next time. If you have any questions, please feel free to leave them in the comments and I will answer them as soon as I can. Thank you very much for watching, and I will see you next time.